Martin for quite a while, and when I spent some time at Inca in the beginning of the century.
Almost.
And it's really an honor for us and a great pleasure to have Martin here. He has been working for quite a while now on T lymphocytes, genetically modified T lymphocytes as cancer immunotherapy. So we're really in for treats today. Martin, thank you very much.
Thank you, Bruno, and thank you all for the invitation and for having me here. So, as Bruno mentioned, we're working with this genetic modification of T lymphocytes for a long time now, more than 15 years. We also do other things in the lab, some biotech focused stuff and some tumor immunology projects as well. But for today, we're going to focus on the CAR T cells or the genetically modified T cells for cancer immune therapy. And this work has been developed mainly on the National Cancer Institute, but also some part of it are starting to be developed also in the FIOCRUZ Foundation. So I brought some landscape of what we're doing. We're doing other things that I didn't bring today, but I would just mention that. So maybe if someone wants to discuss any aspect, I would be happy to discuss this. And please, whenever you want to interrupt me, just go ahead, no problem. So the main problem we're facing here is how to promote the recognition of tumor cells by T lymphocytes, right? And for tumors that are highly mutated, such as this one in the right side of the cartoon, you will find frequently mutations that can become new antigens and be recognized by T cells that are infiltrating the tumor. For those tumors in the left part of the panel, this happens to happen very less often. So it's not expected that you will find many T lymphocytes that are capable of recognizing and responding and eventually eliminating the tumor cell, right? So this is where we are working. For those landscapes where you have a big tumor antigen load or mutation load, you will find very often this kind of situation here. Many T lymphocytes infiltrating the tumor, and many times the tumor will develop mechanisms to counterbalance this recognition and immune modulate the tumor environment. So these T cells will not eliminate the tumor, right? These are the fundamentals for the development of the CTLA-4 and PD-1 blockade that won the Nobel Prize a few years ago. So this is one kind of immune therapy that's very mainstream nowadays, right? For some tumors, such as lung tumors, we can anticipate that virtually all the patients will today, at a certain point of the treatment, be treated with the PD-1 or PD-1 blockade. But here we will speak much more about the other side of the spectrum. So these tumors such as the acute lymphoblastic leukemia that are marked here, and in contraposition to this highly mutated tumor. But again, immune therapy starts always with the more immunogenic tumors, right? So these immune checkpoint blockade molecules are like that, exactly like that. And cell therapy started as well with this very, very mutated fellow here, the melanomas, the metastatic melanomas that had, amongst all the others, the highest mutation load, right? And by consequence, many of the melanoma tumors will have very high infiltration of T lymphocytes. So those will be the tumor infiltrating lymphocytes by definition. And you can find many of them And some groups, such as the NCI group led by Steve Rosenberg, from the 19s on, are just extracting those pieces of tumor, dissociating these tumor biopsies, and stimulating and identifying the tumor lymphocytes that are infiltrating this, the T lymphocytes that are infiltrating these tumors, expanding them in vitro and giving them back to the patient. So those are the TILs, the famous TILs. And for melanoma and some other tumors, such as some sarcomas and eventually even for some breast cancer patients, you can find very important and huge tumor elimination just by reinfusing those T cells that you extracted, cultivated in vitro, and gave back to the patient. So you can see some subcutaneous and visceral metastases, and even this big mass here in the neck that after the tumor infiltrating lymphocytes infusion can even go for a massive necrosis and fall from the regional spot here, and you can see that this patient can be in remission for many years now. So conceptually, this is very important, because if you take enough cells recognizing the antigen, expand them and give them back, you can promote this kind of recognition and tumor elimination. Unfortunately, this is not the case for all the tumors, as I mentioned. So we will speak a little bit about these tumors that have very low mutation burden. And by doing so, they cannot be nicely recognized, and then you have to use other approaches to promote tumor recognition and elimination by T cells. And this is where we get to this different approach that have been tried. All the approaches in red have been tried before for this kind of leukemia, for instance. I particularly worked with anti-CD40 stimulation and leukemia blasts during my PhD. But the fact is that other than bispecific antibodies such as blinatumomab, which is an a BiTE specific for CD3 and CD19, none of the other approaches have achieved any consistent success, unless you consider the CAR T cell therapy that's marked here in red or in black. So this is what we'll be talking about in the next half an hour, maybe. So chimeric antigen receptors are receptors that are chimeras by definition. So the idea here is that you can put together a domain that can recognize an antigen in the surface of the tumor cell, and that could be a protein, sugar, or whatever. You can find an antibody that can recognize, or even not an antibody, sometimes just a piece of a fragment of protein that can bind to something in the membrane of the tumor cell. And then you can assemble this into a new receptor that will be a chimera. So it will be like just like playing Legos and putting different pieces of the Lego final configuration. So you can put a hinge, and then you can use intercellular domains to simulate these lymphocytes, right? So this is very straightforward in terms of immunology. It's something that disregards lots of things that we have learned about antigen restriction by MHC. So pay attention that here you can have one receptor that can recognize the same antigen, no matter the MHC we have here. So it can be used for each one of us, right? So this is one of the features. So it's MHC independent or HLA independent. With high affinity, it comes from an antibody. And even more important, you can use it no matter if you have a previous immune response to the tumor or not. So you can get lymphocytes from the bloodstream, put this transgene into those cells, and they will be anti-tumor cells by definition a few hours later, right? So it's a very straightforward way of making this immune therapy. Of course, to emulate the T cell activation, you provide signal 1 and signal 2. So the CARs were designed first with a signal 1, that's the zeta chain from the CD3 complex. So this will be the chain here that provides the signal 1. Let me just, the signal 1 here. And then you can put some people realize that maybe putting a co-stimulation would make those cells persist longer, be activated in a more physiological way. So what happened was that we had this first generation of CARs and then a second generation. So the first generation is the zeta chain, and the second one has the zeta chain and one costimulatory domain. So see that this is a single molecule, right? It's kind of just putting together different domains. I would never say that this could happen and this could work, but it does, right? And then you have the third generation CAR that along the CD3 zeta has two costimulatory domains, and you can choose among a wide variety of domains. So CD28, 41BB, OX40, CD30, and many others. And then you have the fourth generation CAR where you have the zeta chain of co-stimulation, and into the same transgenic test set, you put another gene. And it can be a cytokine, an antibody, something that would interfere with that microenvironment, make it the tumor microenvironment, making this T cell more capable of dealing with those immune suppressive or immune regulatory environments. So this is the landscape now. We have many products of second generation CAR approved already for commercialization in the US, Europe, and now in Brazil, we have two products approved already. So they started being tested clinically in the 2000s, early 2000s. And this is the first, I would say, success case for CAR T cells, the first generation CAR against ganglioside, that's GD2, for treating child neuroblastoma. So this is very impressive. So it worked at first in solid tumors, and now we have many liquid tumors or hematopoietic tumors being treated, but few cases of success in solid tumor cells. And that what changed the field was were the CARs against the CD19 antigen. So it's a B cell antigen. So, and we will see in the next slide. But the thing here is I want to show you that you can use the zeta chain that's in blue and choose amongst many costimulatory domains. So depending on the costimulatory domain, you can have different fates for those cells, different ways it can be activated, and clinically it can behave different as well. So we have now this CAR that's highlighted here against CD19 with the 41BB or CD137 costimulatory domain. We have the CD28 CAR as well approved for commercialization. And we started seeing very nice results when targeting this CD19 antigen. And CD19 is a very interesting antigen because it's expressed from the early pre-cell precursor until maturely differentiated and fully differentiated B cells. So if this blast becomes a leukemia or later on a chronic leukemia or a lymphoma, whenever it retains CD19 expression, it can be targeted with the same CAR. So again, it can be used for different people, right? And it can be used for different diseases as well. So it's kind of a dream for any pharma company, right? Having the same product that can treat different disease. And this is what happened here. So we have this group from the NCI treating non-Hodgkin lymphomas, CD19 positive. We are second generation CAR, eliminating the lymphoma cells, but also eliminating normal B lymphocytes, because this antigen is shared between the tumor and the normal B cells. So it's a lineage antigen, it's not a tumor antigen by definition. But this product was approved by the FDA. This one was approved too. This is results from chronic lymphocytic leukemia, CD19 positive again. So this patient was resistant to rituximab and bendamustine. So you can see here the blast counts treated with CAR T cells, about 50 million cells. So for the TILs, if you paid attention, it was about 10 to the 9. Here we're talking about 2 to 3 logs less, cells being infused. And here you can have the elimination of about 2 kilograms of tumor in 20 days. And this patient is still in remission. So it's very important. Those cells are living drugs, so they will proliferate, keep memory, stay in the body for many years now. These patients, 10 years later, they still have T cells with the CAR molecule circulating in their bodies. So the way those cells are prepared is a very complex manipulation, and we'll talk about this because this is central for what I'm going to show you. So you can have to collect the lymphocytes, and then you can choose different ways of manipulating and expanding those cells. So you have to simulate them so they can proliferate, and then you will deliver the transgene with a viral vector, many times a retro or a lentiviral vector. So for these vectors, the cells have to be proliferating so they can incorporate in the genome. If you use non-viral vectors such as transposons, then you can make those cells receive and integrate the transgene, even if they're not proliferating, but you have to make those plasmids or whatever get into the nuclei of the cells, and then you have to electroporate cells or something like that. And then you can cultivate those cells with some cytokines for many days or a few days. The protocols can change. Then you will see the transgene being expressed and make all the quality controls. You usually freeze the cells, and then you ship them or take them to the bedside where you can thaw them and infuse them into the patient. It's a very complicated process, but the thing here is that you can change each one of these components, the way you activate the cells, the way you cultivate them, the way the transgene you choose to use, the CAR design, the vector you use, and then the way you freeze or not freezing the cells. So it's just like putting different pieces into a device that you can choose different screens, different antennas or batteries or whatever. So you can make the CAR T cells in several different ways, just like you can make different cell phones with different components. So this is very important. So each group will choose different platforms or different ways of generating the CAR T cells. And this is something we are going discussing now because this is some of the contributions we're trying to make to the field. Again, this is a very complex way, and you can choose, for instance, I mentioned different ways of delivering the cell. So we have chosen many years ago to work with transposons for the single reason that today, to prepare a batch of clinical grade lentiviral vectors, for instance, that you can use in about 100 or so patients, it will cost maybe more than 100, but it will cost about 1 million euros. And you have to wait many times 3 to 5 years in line because it's a big success and all the facilities are booked for the next year. So making plasmids in the clinical grade quality is much easier and cheaper, right? So we decided this a long time ago to use this kind of system. This is a transposon has this funny name of Sleeping Beauty because it was a fossil transposon that has been mutated by reverse engineering until it started to work again. So it's just like, you kiss the Sleeping Beauty and then it wakes up. So you will have the transgene that's flanked by the ITRs, and then the transposase that will cut the ITR domains or sequence and insert your transgene into the genome. So it has been used for some groups now clinically, one group in Germany, one group at the MD Anderson in Houston in the US. So this is the paper for the MD Anderson group. If any one of you want to check the results, and also for the group in Monza where I did part of my PhD. So this same group, by different ways, developed this very similar platform to what we are using, using cytokine-induced killer cells. It's not exactly the T cells were manipulated, but gene modified with a CAR against CD19 and gene transferred by the same Sleeping Beauty transposon. So we have a very good precedent here in the literature and some sister institution doing this. So we have been doing this for a while now. I'm showing here some almost 10 years ago result, a CAR against CD20. So you can electroporate the cells and then expand them, and over the weeks or the days, it will get more and more CAR positive because the CAR cells expand more. So this is a CAR against CD20. So we have CARs against CD20, CD19, first generation, second generation, many configurations. We have been doing this really for a long time now. And what we are trying to do is changing the way we gene modify the cells and changing the way we manipulate them. So making it more straightforward and more cost effective. So we can try to offer this for the public system. So just I work in two public institutions. So the idea here is making this feasible to be applied in the public health system, right? So those are the steps of the CAR manipulation where we are making some contributions that I will show you today. So changing the lenti to non-lenti or non-viral vectors and changing the way we manipulate the cells, right? And I will show you why we're doing this. So I will tell you three short stories, ways of how we are doing and dealing with this challenge. So we get the peripheral blood from donors or patients. We just electroporate them with an electroporator. Now we have electroporators that are clinical grade compliant, so we can use it for clinical therapy. And then we co-electroporate the transposon and the transposase plasmid. So the transposon will carry the CAR gene, okay? They will go to the nucleus, be transcribed, and then the transposase will cut and insert the transgene into the DNA. And from there, we can make different things. We can expand those cells, for instance, using antibodies against CD3 and CD28, provide signal 1, signal 2. Those T cells will expand, and we can use them for therapy. We can also expand those cells using a stimulating cell line. And this is very nice because we can get some cell lines that are Epstein-Barr virus infected. And then we can expand those cells, also stimulating the TCRs that are specific for the ABVs. And virtually all of us have lymphocytes that are ABV positive. So we can end up having a product that can recognize the tumor antigen by the CAR and recognize viral infection or viral antigens by the TCR. This can be very valuable for post-transplantation situations where you immune suppress the patient, and many of those virus will be reactivated. And since we're using non-viral vectors, we can even very straightforward approach, use those cells soon up right after the electroporation. So we don't have to extend them, anything. We don't even have to activate them. We can just electroporate them and put them into the patient. Why not? So I'll show you some results of the different approach. The first one I will show is this in the middle, because it was where we started in collaboration with some groups at the Boldrini Institute and Unicamp, both in Campinas. We expanded those cells for 12 days. And for all the situations I mentioned here, we'll end up testing those cells in vitro for killing of tumor cells and in vivo in NSG immune deficient mice that we engraft with human leukemias expressing CD19. Sometimes this leukemia has also the luciferase, so we can quantify it by just injecting luciferin and seeing the tumor load by quantifying the photons these mice will be emitting. And this is the gold standard preclinical model. After that, you can make all the regulatory things and propose to use it clinically, right? So let's start with this one. We electroporate the cells. The cells can expand or express the CAR molecule here very nicely. We can even put different CARs, combinations, CARs for activation, inhibition. We have done this a lot. But then you can activate those cells with the EBV transformer cell line. That's a B cell that's CD19 and CD20 positive. So it's useful for expanding CAR cells for CD19, for CD20, and maybe some other antigens as well. And then you end up with this situation where you have the T cell receptor recognizing the antigens of the virus and the CAR recognizing the leukemia CD19 antigen. And this is what happens exactly. So if you put a target cell that's both EBV negative and CD19 negative, and you use the cells that were expanded but did not receive the CAR, that's the negative in blue, or received the CAR, this 19BBZ, that's a second generation CAR in red, none of the effector cells will kill the target. But if the target is CD19 positive and EBV negative, in the right panel, you will see that the cells expressing the CAR will kill the target nicely, and the cells not expressing the CAR will not kill so nicely the target cells. And in the middle panel, you will see that if you use a target cells, that's exactly the cell we use to stimulate the T lymphocyte, and it is EBV positive and CD19 positive. T cells will kill the target cells, disregard of having the CAR or not, because the TCR will recognize it. So this is an approach where you can use it for EBV reactivation, and some of the patients that undergo bone marrow transplantation are both minimal residual disease for the leukemia and sometimes reactivate Epstein-Barr virus or virus or many other viruses as well, right? Of course, it has to work also in vivo. So we put these leukemia cells in the NSG mice and treated them with both PBS or the MOC cells. The MOC are the expanded cells without the CAR, or the 19BB zeta cells are the cells with the CAR construct. And you will see that the MOC and the 19BB zeta are capable of controlling leukemia in the circulation, but the tumor infiltration by leukemia cells can be only restrained by 19BB zeta cells, CAR-positive cells, right? And this leads to a very nice improvement in the survival of these animals. So I think we have a very nice protocol in hands. And the plan now is that we're doing already with the Baylor College of Medicine team is calculating whether we can use now donor cells, where we have several bags from donor lymphocytes that would be infused in patients, but for some reason, those cells were not used and are frozen. And in collaboration with the ICB-IBCCF in São Paulo team, we have identified 9 of those bags. They are all HLA typed, so we know which HLA they have. And we also know the HLA of the patients that underwent transplantation and reactivated many of those viruses that are listed here. So we did all the calculation with 7 to 9 of these bags stimulated for these multiple antigens from these multiple or from these different viruses. You can end up having a product that could be used for any patient, for more than 90, covering more than 90% of the patient that have reactivated any one of these pathogens in the National Cancer Institute or at the ICB-IBCCF Transplantation Center. And the reason we're doing this is because when you expand those cells with these peptides from the virus, you restrict in such a way the T-cell repertoire that they do not cause more any graft-versus-host disease anymore, or very mild graft-versus-host disease. So you can use those cells as a bank and use them for them and use them in the patients if the reactivation occurs. With this characteristic of not causing graft-versus-host disease, we think there are very nice off-the-shelf platform we can use to put the CAR T cells as well for treating some of those patients that will have minimal residual disease as well, right? So this is a project that we're running that's funded by the CNPq, and the idea is exactly this. You can have the T cells that can recognize by the TCR. These different clones would be mixed in this product, each clone recognizing antigens from the pathogens. So the only match you have to have is the MHC or HLA allele that will present the immune dominant peptide. If you have 2 or 3 of those alleles, even better. But with this calculation, we can cover more than 90% with 1 allele at least and 70% with at least 2 alleles. And by putting the CAR, you can recognize now the tumor antigen. The second approach is much more straightforward. So we just electroporate the cells and activate them with this mix of antibodies. And this more canonical way, it's the way people is doing all the CAR T cells are approved already for therapy. And then we have from a very short protocol, it's 8 days. The other one is about 2 weeks. And so it's a short protocol, cells get enriched in CAR expression over the cultivation period. We will end up having basically T cells in the culture, so that's expected. You're simulating selectively the T cells. And a very nice thing here is that you had a very cool central memory phenotype at the end of the expansion. So those cells are the cells that are capable of, once in vivo, support the differentiation and constant import of effector T cells and sustain this response longer, right? If you have some terminally differentiated T cells, it's good for first killing of the tumor, but not for sustaining this response. So we have this very nice phenotype. These are published data, the one I showed before and this one, so you can check the paper in details if you want. They will kill the tumor in vitro in a very nice dose-dependent fashion here in A. They can kill different leukemia, CD19 leukemia cells, and produce gamma interferon and many other nice cytokines, such as in C and D here. And of course, it has to work in vivo. So we have now the tumor with the bioluminescence. You can quantify it. And you see that the animals treated with the PBS here in C in the left have a very nice tumor load that goes increase and it's quantified here until the animal died, because I mean the leukemia kills it. So we have the survival curve here in G going down for PBS, the same kinetics for mock electroporated cells. So cells that were cultivated the same but did not receive the CAR. And if the cells receive the CAR, you see a very nice control of the leukemia over time and a very nice much lower bioluminescence and much better survival. So this is a straightforward protocol that we're proposing. It's funded already by the Ministry of Health. So we'll take it to the clinics soon by making a phase 1, phase 0 trial. So it's a feasibility and toxicity trial for 3 patients with acute lymphoblastic leukemia and 5 patients with B-cell lymphomas. So this is something we're very delayed now because of the pandemic. So we're trying to catch up with the original timeline to see whether we can start treating the patients next year, probably. Still, those protocols I show you are very complex in terms of you have to cultivate the cells a long time and have a very important and running facility so you can have all set to produce those cells. And we know that by electroporating cells, we can electroporate them later on after they are activated, or we can even just collect the cells and electroporate them. So you could have those transgene cells right away in a few hours, even minutes maybe. And we also know that the more you expand and cultivate those T lymphocytes in vitro, the worse they perform in vivo when you put them back. Many times they are kind of skewed or terminally differentiated or exhausted. So the less you cultivate them, the better they perform. And this has been shown by several groups. One of the groups that has shown this is Michael Mylan groups in UPenn, showing that if you cultivate those cells for 3 days instead of 9 days, they can control leukemia much better. So the less you cultivate them, the better. The problem is that by using lentiviral vectors, you have to at least cultivate them for 3 days, maybe an overnight, so they can enter cycle, get activated, and then incorporate the transgene. The electroporation system has the advantage that you can maybe just take the cells off, gene modify them, and put them right back into the patient. Maybe wash the cells because you have lots of plasmids around, but you can put it right away into the patient. So let's see how it works in the animal model, the same animal model. So we can take the human cells, lymphocytes from donors or patients, electroporate them, and put them right away to the animal. So this is what we call the point of care, because it could be done even at the bedside, right? So you don't have to take this to any facility as soon as the system is closed system, okay? So this is the animal model. It's very similar to what we showed before, but here you will electroporate the cells with no plasmid or with the 19 BBZ plasmid, or treat the animals with the PBS. right? So we let the cells rest for a few hours because electroporation is very aggressive, so the cells can recover for the day, 4 hours. Or maybe if we want to see the CAR being expressed, we can wait an overnight so the transgene gets expressed. But at 4 hours, you don't see any expression of the transgene yet, right? But if you wait overnight, you can see, and it's also published, it's a Williams up your work. So if you wait an overnight, you will see the expression of the CAR here. And if you treat the animals with the PBS, tumor load will be very high in blood, bone marrow, spleen, liver. The mock can lower a little bit of the tumor load in some organs, but not in others. And the 19-BB-zeta will very nicely control the tumor load, providing a very nice survival curve for those animals. So it works, right? And it can be used, and it could be much simpler than expanding and cultivating those cells. Of course, it works, as you saw, but let's compare them to the more canonical protocol of cultivating the cell. Let's see if it works at least as well as the expanded cells. So again, you cultivate those cells, you will end up with this central memory phenotype that's very nice for the cultivated, the activated cells. And then you can have here for the same number, and here we put very few cells, very low number of CAR T cells because we wanted to amplify any difference. If we put a very high number of CAR T cells, you can even virtually cure the animal and see no difference. So these are very low dose, very, very low dose. And then you can see that in the upper panels is the point of care approach, in the lower panels is the expanded cells, and they behave just the same in terms of capability or capacity of controlling the leukemia, especially at the highest dose, that's still very low, and provide a benefit in terms of survival. If something, the point of care seems to be slightly more potent in controlling the leukemia, even at very low cell numbers. So I think this approach can be used and can be very much more straightforward than cultivating the cells and keeping them in culture for many days and whatever. This is a platform, so you can put the CAR and then you can put many different things together with the CAR. Remember the 4th generation CAR. So we are testing some things as well. I didn't bring much of it to show you because it's still very preliminary, but one of the things you can do is put the membrane bound IL-15 to the cell so it can be stimulated by the IL-15 receptor, and those cells can get much more independent from outside the stimuli and behave better in the tumor microenvironment. And by doing so, you can see here that you can have, so this is tumor only growing in red and black, mock in red, 19-BBZ in blue, the classical CAR we're using, and the CAR with the IL-15 here in the last panel, so you can see we have a better control of the leukemia. So you can potentiate this anti-tumor effect by putting different things in the same cassette you're transferring to the T cell, right? So this is a part of some of the projects we're developing, putting different molecules along the 19-BB-zeta CAR. So we have these 3 initiatives now: the EB-Vila cell line, simulated CAR T cells expanded that are bifunctional. Now we are just further developing this for multiple specific, so the VST, the virus specific T cells or multiple VSTs as a off-the-shelf platform for CAR T cell. We have the expanded cells showing anti-viral activity, so this will be our first clinical trial. And we have this point of care strategy that we know now that they have similar potency or at least a slightly higher potency against the tumor, and it can be game changer because you can, for instance, if you have a facility and you have to occupy it for 2 weeks to prepare a cell, we can prepare 2 patients for a week, but if you can do it overnight, you can prepare 20 plus patients per month. I mean, 2 patients per month versus 20 or even more. So I think this could lower a lot the cost of generating those cells. And then you have new car developments, and I will use my last 5 minutes just telling, we're doing some other things I will not bring, but just to give you a flavor of what's going on in the field. So there's a lot of hype about NK cells with CARs. So those will not be CAR T cells, will be CAR NK cells. These are very nice. It's a small trial yet, but showing that you can have similar response rate with much less toxicity. One of the complications of CAR T cell therapy is the cytokine release syndrome, but those NK cells do not promote this kind of adverse event in such a high frequency. So there's a lot of hype. We have been doing NK cells for a while. We stopped a few years ago. It's a very interesting protocol. We have different things, such as the idea that you can change this model from very personalized therapy, where you take the cells from the same patient and prepare it to the same patient, to a model where you can use somewhere else patient cells and use for different patients. So I show you the VST platform as an off-the-shelf, and there's a different way of doing this. That's by gene editing and taking donor cells from a healthy donor, whoever it is, and just shutting down or knocking out the TCR receptors. So we have some a project running in FIOCRUZ with this. So you have to distinguish this feature that makes the T cell a T cell. That's the TCR, right? So you can, by telling CRISPR or zinc finger nuclease, eliminate the TCR. You can eliminate other things as well, such as the HLA and other receptors, and put now a new specificity in this cell by the CAR molecule. So this is something we're pursuing as well. I didn't bring any result, but this group in the UK has treated some patients now with this kind of off-the-shelf gene edited T cells. So it's very nice work. It's been improved, but it's the idea that you can generate this bulk product that can be frozen in fractions, and then you can use one of these bags for each patient. So the paper is here if any one of you want to see the results. And then there are some things that can happen that would make this therapy even faster and cheaper and more straightforward. That would be maybe delivering the cargo for the transgene directly into the patient. So there are some initiatives using viruses, lentiviruses with some modifications for the tropism, so they can infect T cells. And there are some other initiatives using some nanocarriers or something like that, that you, and even a recent paper using some just like gels where you load the T cells with the viruses and put it subcutaneously into the mice. But here you can see that delivering in vivo this cargo with the transposon as well can make the T cells express the CAR and eliminate the leukemia that's into the organism already. So it's very straightforward, no cultivation, no ex vivo. So this is another tendency we're seeing running. And to end, I think we have other application of CAR T cells. So the title was about cancer immune therapy, but I think this is a very nice platform that can be used to eliminate any cell you want, as long as it has surface markers. So here there are some 3 examples of this that have been recently published. Some cells, CAR T cells to eliminate senescent cells, some senolytic CAR T cells, recognizing this UPAR receptor here. You can have CARs eliminating fibroblasts in cardiac lesions. That's a paper that reverses partially the fibrosis in the heart and makes a gain of function by targeting this fibroblast activated protein. And this last one is very nice one. Again, the configuration of the CAR, that's not a single chain fragment variable antigen recognition, but a fragment of the antigen that's targeted by some anti or autoimmune B clones. Right? So you have the target for the BCR of those clones, and once it interacts with the T cell gets activated and eliminates selectively that B cell clone, getting rid of the. It's much more cleaner than using, let's put rituximab to eliminate all the B cells if you have lupus or something like that. So many things can be done with the CAR T cells, just to give you a flavor. And of course, for infectious disease as well, you can target many different targets that get expressed in the tumor, in the cell surface, the infected cell surface. So CARs for HIV, HCV, HBV, CMV, EBV, even fungus, right? And you can even treat some autoimmune disease by redirecting some Tregs towards the tissue by putting a CAR that will recognize an antigen that's specifically expressing that tissue. So some examples here. This is a review we published last year. This Mariana Mazzi, it's our PhD student in our group. So if you have interest, you can go through it. So I think it's a platform where you can use different approaches to kill and eliminate different cells, or even to suppress or change the immune response in different tissues, right? So we envision already that maybe we will have many of those things running soon in different protocols. So I just end up by acknowledging the people here in my lab in the left column in red that generated results that I showed today. It's all published, on most of it, so you can go through the papers. Our collaborators from different institutions for the results I showed today, and the funding agencies, of course. And if you want to check out the publications and some things, that we have a blog in our site where we discussed many of the publications that are coming out these times, and our own publications and many other groups, nice works. You can go to the page in our from our webpage, where you can find the blog and the list of publications. So it's all there. And I will be very happy to take any questions if you have. And thank you again, Bruno, for inviting me, and the people here for your patience and your time. Okay, thank you very much.
Right, Martin. Thank you very much for a very nice talk. We usually first open the floor for questions from the students. So, do we have any questions, Lucas? If you could come closer, so come closer to the microphone.
Thank you so much. That was an amazing presentation. I actually have two questions. The first one is, can you also induce the expression of some lipid mediators associated with CARs, so that you can deliver some lipid mediator to a specific receptor, I don't know, to directly to the tumor cell?
Okay. Yes. So I think those cells are platforms for what I showed here is 1 gene, 2 gene modifications, but we have a whole field of synthetic biology running with very nice things being done. So we're part of the synthetic biology INCT initiative, and some of our work deal also with generating system for regulating the expression of 1 gene, 2 genes. But some groups are developing even the whole path or metabolic path for things. Recently, opioids, and then you can. So as soon as you have a cargo that's large enough that can be carried into the T cell, then you can envision that you can put maybe some modulators of, let's put any inflammatory mediator or whatever you want, or even you can put some orthogonal independent biosynthesis pathway as well, right? So this is something that hasn't been done already in such a refined way, as you're mentioning, but it's possible and it's getting more feasible every day. So many groups are working in the cargo with especially non-viral vectors for larger cargos. So you can put, because if you have to put 3, 4, 6 genes, it doesn't fit anymore, either in the sleeping beauty or in the antiviral retroviral vector. So we have to go into piggyback sleeping beauty, piggyback transposon, or other systems. And there are some very nice systems with plasmids that replicate independently, so they can keep the expression even if they don't integrate. And this could be very important for safety reasons as well, right? So, and with this system, this kind of thing you mentioned are very feasible, I think.
The second question is, do you predict that in solid tumors specifically, the T regulatory cells might be an obstacle for this approach?
Definitely, definitely. So for CAR T cells, solid tumors are something that we have struggled a lot to. And now we're starting seeing some anecdotal reports or things that start working. One of the last we saw was IL-15 together with the CAR. Some results in the literature show that if you put IL-12 on the CAR, or the CAR T cell produces IL-12, it gets refrigerant to or resistant to the Tregs and to my microvirus. So we have different ways of approaching this, but definitely myeloid cells or mature myeloid cells or Tregs and many other results or systems or circuits for immune modulation are very important, different for each kind of tumor, right? And then you have to approach it this way. But Tregs definitely are one of the main players.
I focus it on Tregs, but I believe that myeloid-derived suppressor cells might be a problem.
And many other, even soluble molecules of IDO and other TGF beta. There are very nice results also with dominant negative TGF beta receptors to get the cells refractory to TGF signaling. So many ideas. Nowadays, anything you can imagine, or almost anything you can imagine, is being done by some fellow somewhere, you know. But definitely, yes, the regs are important.
Thank you.
Thank you.
Chris has a question.
Thank you very much for your expressive thought. So I just want to ask you if you can put like CAR T cell, CAR receptors in macrophages, or
Yeah.
so you mentioned NK cells, and I have seen some studies
Yeah.
NK cells. I heard that I saw some studies in macrophages. So can you infuse macrophages and CAR T cells to, maybe regulate the suppressive microenvironment? Can you put like chemokines to drive macrophages?
Exactly. So that's a very good point. I didn't mention macrophage and should, but I thought it was already too long. But one of the key obstacles for solid tumor is homing of those cells, right? So the good results we're seeing, the first one, where you inject the CAR T cells straight into the tumor, right? So you can, overcome or disregard the homing issue. In the macrophage, home very nicely to the tumor. So that's why some groups are doing this CARMA or this CAR T cell, this macrophage with CAR molecules. And macrophage can be very nice in phagocyting and also killing tumor cells as well, right? So the idea is exactly that. So they will migrate very nicely and get activated by the CAR and kill the cell. So there's even a company that's doing this and infusing the first cells few months ago. And there is also an initiative of generating those macrophages from IPS. Let's put it this way. So expressing the CAR as well. So the whole field, again, anything you imagine, probably there's some guy doing this somewhere. But let's keep thinking. So maybe we can find different ways of doing this. But certainly macrophages are very nice targets as well. Yeah.
Okay, we have another question from a student here. Gabriela. And also it's more of a technical question, and I will actually add up a question that I have. Is that Gabriela's question is, what would be the method for inserting these cells grown in vitro back into the patient? Is it like a vaccine? Is any adjuvant needed? And also would like you to comment how effective is the electroporation and how many cells you can actually make as a CAR T cell.
Okay, so for the first part is you just put them intravenously and they will home especially for leukemias and lymphoma, they will just go around and find these clones. So this is the way it's done. For solid tumors, as we just discussed here, sometimes it would be nice to inject them inside the tumor if possible, right? And this is where we're seeing some nice results in neuroblastoma and some mesothelioma, so we can put it into the lung already. But certainly this homing issue will be addressed, and it's being addressed by several groups just by putting some chemokine receptors or something like that. So this is point one for the electroporation. So you have different electroporation systems. Some of them you can adjust more, more refined in the settings, and then you can use different ways of doing this. Some of them will keep the cells alive in a big proportion. Some of them will kill lots of cells. And then you can change also the conditions. We're using plasmids, but there is another group in Germany using the transposase as a protein or RNA, and the donor plasmid as a mini circle that says only the function of part you want. So the less DNA you put, the less toxicity you have. So you can make all the adjustments. In our hands, we have electroporations that if we evaluate by, 24 hours later, it can get from 6 to 30% positive. It depends a lot on many things. And we're working on optimizations in this aspect as well. You have a massive cell death, but if you expand those cells, at the end, you have beautifully, by day 8, as I show here, most of the cells will be T lymphocytes, very nicely activated, well and alive. For the point of care, this could be critical, and then we're working on. But for both situations, our calculations that we can get enough cells to treat the patients with the regular dose that will be about 2 million CAR T cells per kilo. And then again, since you are electroporating cells, it depends on how many cells you are electroporating, right? So you can electroporate up to 1 billion cells in one device we're using. And then if you get 10% of them alive and well and expressing the count, it will be more than enough for these doses I was mentioning here. So you have just to take the chance and make all the calculation, but it's feasible and we're working on this. So.
If I recall correctly, when you started doing this, there was all these concern on having some sort of kill switch for these cells.
Yeah.
So if you had any trouble coming from the insertion of the genes in these cells, you could use this kill switch to get rid of the treatment. Exactly. That was now
not only with the insertion, but also with if you take the CD19 as a target, it's a very nice target, but you still target the normal cell, right? And then you can mitigate this by fusing immune globulins, right? No, so it's not life-threatening, but in other situations it can be if you choose other targets. So it would be much more important for this context than for, let's put it, potential insertional mutagenesis. Mm-hmm. So now we have thousands of patients treated with lentiviral vectors, and we have very few reports, especially one report of one patient with chronic lymphocytic leukemia. It was a patient of the 6th decade, and it's very common to have a skewed hematopoiesis because you have mutations in some epigenetic regulatory proteins such as TET2. And for this patient, he had already a TET2 mutation in one allele, and in one of the cells transduced, the CAR construct landed in the second allele. So this clone expanded a lot, but did not transform, killed the tumor, and then it shrank as a clone, and it was okay. So maybe it's a good place to put the transgene, right? And so this was the readout. But we have a recent report for one group in Australia using a different transposon, that's the piggyback, where they treated few patients, and 2 of them, at least, if not 3, developed lymphomas on the cells carrying the CAR. And they publish these results, but it's not really clear what happened because it doesn't seem to be a classical mutation insertion or mutagenesis mechanism. So it kind of obscure, maybe the mechanism will come out, but nothing yet for the sleeping beauty as adverse event. And for lentium and retroviruses, so far it is running well. So the suicide genes will be more expected to be used when you use a card to recognize something that you don't know exactly what is expressed in the cell or in the body in all the situations, because expression can change whether you're inflamed or, and there is no good preclinical model. So how can you evaluate this? If you put it into the mice or into the mouse, it will not express the human molecules in the same fashion humans do. So this is a situation where you can use a suicide gene, and for those largely gene-edited cells, maybe as well. I showed the TCR knockout, but there are some groups making knockouts for multiple immune checkpoint receptors, such as PD-1, CTL4, and then the Pennsylvania group did a trial with 3 different knockouts in the same cell. So can be a big mess, right? And then in this situation, maybe suicide switch would be required.
Right. I guess we have a question from São Paulo, Gustavo. That's you. Or
Maybe
Rita?
maybe I can stop sharing so we can see them.
You already stopped sharing, Martin.
I stopped sharing.
Yeah.
Okay. I have to stop here. Okay. Maybe exiting the presentation.
Yes.
Okay. Let it this way.
But you have to put the sound output of the computer to the TV.
Okay.
It's using the internal speakers.
Can you hear it? Okay.
Gustavo?
Hey, Gustavo.
Hey, can you hear me? Can you hear me?
Yes.
So, Martin, yeah, so very, very sorry I missed most of your lecture. I was caught in this committee to recruit students to our program. So I have a couple of questions. I hope you haven't mentioned that before, otherwise I will look stupid here. But anyways.
Never stupid, Gustavo.
So there's a couple of things that I'd like to ask you regarding this CAR T cell strategy. One is the importance of making these cells long-lived memory cells. So I think this might be one of the things of making a T cells or CD8 T cells as a CAR T cells, and whether the CAR receptor would trigger signal to make the cells now expand versus you need to rely on the activation endogenous T cells.
Okay.
So this is one point. Can you comment?
Yeah. So one of the slides I show is that you can use several different ways to cultivate and expand those cells. And definitely one of the issues now is when you expand those cells is the cytokines you choose to use, right? So there is a shift now from originally people using IO2, and now many groups are using IO15 and IO7. So just with this idea of keeping those cells less differentiated and not expanding Tregs and so all these aspects. The other thing some groups are pursuing is small component or small compounds that can be used to keep those cells in a stem cell memory phenotype. So, and sometimes you do this and then cells will expand much less efficiently in vitro. But then the only way of checking whether this is effective or not is putting them in vivo in this mouse model. So sometimes much less cells will perform much better in vivo if they have the proper or the right phenotype, right? So this is a huge field. Some groups are doing contributions and using this, both during the cultivation and also as many groups and ourselves as well are doing, just putting some of those receptors or cytokines of signalings into the cassette that will be delivered to the CAR T cells. So we have some cells now expressing IL-15, membrane-bound IL-15 that will signal to these cells to make them persist more and achieve a better phenotype once, especially once in vivo, right?
Yeah, that's my confusion, because I think this is regarding the in vitro expansion. But once the cells are delivered to the patient, so do they really differentiate it into memory cells?
They do.
And if they do, what would waken them up, to fight tumor, in the years to come?
So it depends a lot on the context, Gustavo. So they do have memory cells. There's a recent paper from those early CAR T cell treated patients 10 years ago. Those cells are still there. And surprisingly, they are T cells with a very strange phenotype. Right. So there are lots of things on how you assemble this CAR molecule and the signals you choose that make those cells go away a path that's maybe not exactly the canonical path a cell that's activated by its TCR will go through, right? So we're just learning now what happens. And the other thing you can do is changing the co-stimulation or the different domains you have in the CAR. Some groups, for instance, have shown that if you put the co-stimulation not in the CAR molecule itself, but in trans, it can work more physiologically and the cells will perform better. So many things are being tried, right? And your question is very important. And the answer is we're just starting to learn in the long term, how do these cells behave, right? And what's the fate of those cells after, let's put months or maybe years. So it's a very fair question. And the answer is we're just starting to learn about it.
And the other thing, Martin, is, as I understood towards the end, that's the part I watched your lecture, is new cell types, such as the NK cells, as delivery of the CAR T cells. So apparently, as I understand, this is a more acute, so it's a little bit like a tool that you could use to acutely fight, but then the cells would disappear from the hosts. Is that the idea, or?
No, it depends a lot. Again, for instance, the two products that are approved are CAR T cells against the same antigen, but they use different co-stimulation, CD28, 41BB. In vivo, in the patient, the CARs burning the CD28 molecule expand much faster, but persist much less. The ones with the 41BB ramp more smoothly or slowly, but they persist much longer. So if you have a very acute leukemia, maybe you will need something to hit them hard in the beginning. Otherwise, you will lose the run, right? So it depends a lot also on the type of tumor or disease you're treating. And again, for some situations, if you get rid of the less tumor cells, getting rid of the T lymphocytes later on could be beneficial, right? We're just discussing this with Bruno, maybe with some suicide gene, or maybe just by letting the cells. But pay attention that for the CD19 antigen, it's very difficult to do this because you have B cell precursors popping up from the bone marrow every day, right? So we'll keep engaging the CAR molecule. This is not true for some antigen that would be exclusive from the tumor. And once you eliminate the tumor, you will end the CAR cell, the CAR molecule engagement, right? So it depends a lot on the context.
And the last question, Martin, is a different approach comes from inoculating it's a mouse model, of course, but inoculate the mouse with some attenuated pathogens, which would, just increase perhaps the innate immunity or the whatever nature of the microenvironment, the suppressive effects of the microenvironment. So do you think that for the solid tumors, would this help the cells, the CAR T cells, to find the tumor microenvironment and maybe act combinedly? And is this something that is already being tried?
Yeah, so it's a very good approach. There are some, we have a project for CAR T cells and oncolytic virus that's a little bit in line with this idea, and many others have some similar ideas. The problem here is that if you want to do this in a mouse model, then you have to go fully mouse model, right?
Yeah.
So if you use the NSG, for instance, the cytokine release syndrome, that's the main adverse event. It couldn't be anticipated from the mouse models because you do not have a crosstalk of the mouse macrophage with the T cell, and this is the mechanism for amplification of this in vivo in patients. So you have to make humanized models or go fully mouse models, and then the antigen is not exactly the same. So it's not that easy, but many of those things are done, have been tried with, especially with oncolytic viruses and immune checkpoint blockades, many association with different things, and some trials or some projects and publications also with some vaccines and pathogens as way of boosting the innate arm of the response as well, right? So you're right, but it's a little bit more challenging in terms of preclinical model. That's why some groups do not do this.
Yeah. Cool, man. It's great to see you. Keep up the great work. And I'm really sorry again that I missed, I was very late.
Thank you, Gustavo.
All right, great. I actually have one more question. I know it's not what you have been working on, but it's regarding the other side of the equation. What strategies are being employed to identify antigens on the tumors that are amenable to this sort of treatment?
Okay, so it's a hard one. Many groups are doing this kind of differential. So the main path would be do this differential expression of different transcripts, and then you can filter for membrane things. Mm-hmm. So this is the main limitation of CAR T cells. So you have to do the recognition on the membrane, right? And some other groups are doing proteomics and other omics. Some groups, such as groups even here in the UFRJ, trying with glycobiology. So whenever you have something that's interesting in the membrane, people want to try to do a CAR T cell just by the main pipelines will be these omics, both proteo or transcriptomics, and filtering things that should be in the membrane. And sometimes you get very, not expected or unexpected things, and those are maybe the best. For instance, this senolytic CAR was exactly that approach. Senescent cells, what do they have in the membrane, transcripts, and then test it's clear, it's not there, selective, and then make a CAR. So this will be most of the works will be based on this kind of pipeline to describe it.
Great. If we don't have any more questions for Martin, I just want to, let me open here, announce next week's conference would be by Professor Elena Lansuskaya from the University of North Fluminense. Title: Evolution of Environmental Mycobacteria in True Human Pathogens. focus on Mycobacterium kansasi. So next week, same channel, same time.
Okay, thank you.